Hello everyone, hope you all are doing well. So today we are going to do topic 56 of paper 2, and here we will be discussing seed development, germination and seed dormancy. So let's start this session. So first topic we are going to discuss here is stage of seed germination. Or you can say physiology of seed germination. Both are similar terms. Here we will discuss certainly in stages of seed germination. We certainly discuss some physiology only, nothing else. So the first stage is imbibition. It is the absorption of water by dry seeds. It is nothing but absorption of water by dry seeds. So what happens here? Seed coats rupture. Seed coat ruptures. Now second step is respiration. So after imbibition, resumption of metabolic activity. So at imbibition done, there is resumption of metabolic activity. And what respiration is? It is a metabolic activity. Resume the metabolic activity of seed which was not there. It was previously in the previous plant. Resumption of metabolic activity. Initially anaerobic. That is due to energy provided by glycolysis. It is due to energy provided by glycolysis. As in the start, the seed is not able to respire properly, so it is only not able to take oxygen properly, so it is normally anaerobic. Then it becomes aerobic as oxygen starts entering cells, or you can say entering cells of seeds. As the oxygen starts entering seeds, as the initial phase the oxygen was not able to enter seeds, so it was anaerobic and the energy was provided by glycolysis. But as oxygen starts to entering seeds, it becomes aerobic. Now there is certain effect of light. We have discussed how red light, or you can say blue light, enhance germination, or sometimes they inhibit germination also. So there is proper effect of light. Number 4, there is mobilization of reserves and role of growth regulators also. Mobilization of reserves and role of growth regulators. GR means growth regulator. We have discussed plant growth regulators in previous lectures. You must see it, very important. Here, stored starch, protein or fat need to be digested. Stored starch, protein and fat need to be digested. Digestion starts, so they need to be digested. Giverlic acid initiates synthesis of hydrolyzine enzymes. So here giverlic acid, that is GH, giverlic acid, gibberellins comes from gibberellins, initiates synthesis of hydrolyzine enzyme. Now the next step, that is number 5th, is development of embryo axis into seedling. into seedling, embryo becomes metabolic. That means embryo becomes metabolically active. Now from a figure you can also state how germination process is. So seedling and seed, water content. So here we'll discuss how water content increases in seed and how development process starts. So first of all, there is, let it be a graph like this. First process is here mitochondrial synthesis. Mitochondrial synthesis starts. Second process, transcription and translation of mRNA. And translation of mRNA. Third, there is hormone biosynthesis and deactivation. This step there is hormone biosynthesis and deactivation. And then at this stage there is embryo weakens and testa ruptures. At this stage embryo weakens and the testa ruptures. Here there is radical emergence, radical, that is which becomes root, radical emergence. Then there is reserve mobilization, reserves starts to mobilize, reserves mobilization. And at last there is seedling growth. So this is the whole process. Through this type of graph you can discuss how water content increases and this process takes place. So this is the figure showing germination process. Hope you all understand this. Now moving on, what is the difference between orthodox seeds and recalcitrant seeds? Orthodox seeds and recalcitrant seeds. What is the difference between two? So the main difference is that orthodox seeds tolerate extreme drying. These are those seeds that tolerate extreme drying, whereas recalcitrant seeds, they do not tolerate extreme drying. That is, they can tolerate drying up to 2 to 5% of weight. That is, the moisture levels decreases to low moisture levels. It decreases to 2 to 5%, whereas the moisture level in recalcitrant seed is around 12 to 31%. If the moisture level reduced to this, then the seed will start drying. So they are as they are tolerant, they are intolerant to drying. Here in orthodox seeds, longevity increases as moisture levels and temperature decreases. Longevity increases. That is, these seeds can be stored for long time, okay, at low moisture levels, and it is also tolerant to low temperature. But this is recalcitrant seeds are intolerant to low temperature. Hence they cannot be active for long time, whereas orthodox seeds are active for very long time. That is, as they are tolerant to low temperature, they can be frozen also. But recalcitrant fluids cannot be frozen. They are often long lived. That is, they can they can be stored for long time, and they lose viability rapidly after ripening. So you often have seen some seeds, such as example you can say jamun seeds, mango seeds, lychee seeds, jackfruit seeds. These seeds are not able to be stored when they need to be grown, or if they are not grown in certain times and their moisture level decreases, they get dried up, they die. But the seeds of many plants such as guava, apple, these are orthodox seeds. They can be stored at low moisture content for longer time. They are often long lived. So this was the major difference between orthodox seed and recalcitrant seed. Now moving on to the next topic of the session, that is seed dormancy. Seed dormancy. It is the state or condition in which seeds are prevented from germinating. In which seeds are prevented from germinating. Prevented from germination. We prevent seeds from germination. It is an evolutionary adaptation. Evolutionary adaptation that prevents seeds from germination. That prevents it during unsuitable ecological conditions. During unsuitable ecological conditions. That would typically lead to a low probability of seed survival. That will typically lead to low probability of seed survival. So it is an evolutionary adaptation that prevents seeds from germination during unsuitable ecological and unsuitable conditions, in unsuitable and that types of unfavorable conditions, in which if they germinate they will not survive. So they adapt themselves so that they only germinate in suitable conditions. So what are the reasons, or you can say causes of seed dormancy? They are light, temperature, hard seed coat, period after ripening, period after ripening, germination inhibitors, immaturity of seed embryo, immature seed embryo, immaturity of seed embryo, impermeability of seed coat to water and oxygen, impermeable seed coat, impermeable seed coat, that is seed coat not allows water and oxygen to enter the seeds. There is no exchange of water and oxygen. Mechanically resistant seed coat, mechanically resistant seed coat, presence of high concentration of solutes, presence of high concentration of solutes. So these are the factors, or you can say regions or causes that leads to seed dormancy. high temperature, germination inhibitors, impermeability. There are many causes. You can know some of them or you can know all of them. So these are the reasons or causes of seed dormancy. Now, what is the physiological basis of seed dormancy? Physiological basis of seed dormancy. First one is GH, that is Giberlins and auxins. That is growth hormones. So Giberlins and auxins are these two growth hormones that induce dormancy. Induce dormancy. There are germination inhibitors. There is immaturity of seed embryo. So these three are physiological basis. These are related to physiology of seed. While rest such as seed coat, mechanical resistance, etc., which we discussed in the regions above, are physical basis, are due to physical reasons. But these three, growth hormones, germination inhibitors, and immaturity of seed embryo, are physiological basis in the regions or even in the causes. Now, there are two kinds of seeds: dormant seeds and hard seeds. So you must understand the difference between the dormant seeds are those seeds that are alive but do not germinate. Alive but don't germinate. That is because of these physiological regions or some physical bases, physical regions. But hard seeds, they are type of dormant seed that has a seed coat impermeable to water. They are a type of dormant seed, but here the main region of dormancy is this hard seed coat, hard coat. This hard coat, what is? It's impermeable to water, or you can say water and oxygen. So water is not able to go into the hard seed coat, and therefore the seed becomes dormant. So this was the seed dormancy. Now, how can we break seed dormancy? We need to break seed dormancy if we want the seeds to germinate. So how can we break seed dormancy? Methods of breaking seed dormancy. Methods of breaking seed dormancy. First one is natural methods. That is, when seed get appropriate environment, when seed gets suitable environment, then naturally break dormancy. So natural methods are rupturing of coat due to microorganisms. Rupturing of coat, which leads to a movement of water and oxygen. So it can be due to microorganisms, temperature, but as due to high and low temperature, density of seed coat increases and they sometimes break. That could be a region, animals could be a region. So these are some of these can lead to rupturing of seed coat. There is competition of over ripening period. Leach of seed coat inhibitors. As the seed coat inhibitors which were inhibiting germination leach out from seed to the germinate. Inactivation of inhibitors. Inactivation of inhibitors due to light, temperature, etc. Leaching of excess and highly concentrated solutes from seed. Leaching of excess and highly concentrated solutes from seeds. Production of growth hormones to neutralize effect of inhibitors. Production of growth hormones. GH means growth hormones. They neutralize effect of inhibitors. So these were some of the natural methods. Now moving to what are artificial methods. Artificial methods, these are action of hot water action, mechanical rupturing of seed coat, the exposure to heat, cold and light, simultaneous exposure to, you can also say heat, cold and light. We will give heat for some time, then cold for some time, then we will extreme light or low light for some time. Apply hydraulic pressure for 5 to 20 minutes. Hydraulic pressure on seeds for 5 to 20 minutes depending on hardness of seeds. Treatment with concentrated sulfuric acid. So these are some artificial methods, and elaborating it, there are treatments, there are several treatments to break dormancy. We can also discuss this. Treatments to break dormancy. So first one is seed coat treatment. Here to make seed coat permeable to air and O2 by softening and cracking. Here we will do soften the seed coat or cracking the seed coat. Softening or cracking. This is known as scarification. To give a scar, form a scar on seed coat. That will help in movement of oxygen, air, water, everything. Then we have got some embryo treatment. That is stratification. There is scarification, it is stratification, it is known as low temperature. And we have also high temperature treatment up to 40 to 50 degree Celsius. Now third one is chemical treatments. Here we can use mainly PGRs. We use here plant growth regulators. I have discussed PGRs in detail in previous lectures. You must see it. Now moving on to the next topic of this session, that is polyembryony. Here we are going to discuss polyembryony. So occurrence of more than one embryo. Poly means more than one, and embryony means occurrence of more than one embryo in seeds, which results in emergence of multiple seedling. Embryon seeds, which results in emergence of multiple seedling. Now, what are the causes of polyembryony? The major causes are differentiation and development of various maternal. Major cause is differentiation and development of various maternal and zygotic tissues associated with ovule of seed. So crop showing polyembryony are. So this is differentiation and development of maternal and zygote tissues associated with ovule of seed. The crop showing polyembryony are citrus, mango, jamun, almond, rose apple, etc. Now what are the significance, or you can say advantages of polyembryony? Let me rub it. Significance / advantages of polyembryony. First of all, it obtains true to type seedling obtains. True to type seedling. Secondly, reduced cost of seedling production. There is reduced cost of seedling production. There many seeds can be produced at less time. Embryo developed from nuclear embryo provide uniform seedling. As it is developed from nuclear embryo, it gives you uniform seedlings. For root stock, uniform seedling for root stock, which yield constant resulting fruit production. Yield consistent result. Consistent result in fruit production. Nuclear seedlings have better tap roots and has a root stock develop better roots. So they have better roots, better tap roots, or you can say better root stocks. So what nuclear seedlings have? Nuclear seedlings have better root stock or tap root. Nuclear seedling restores vigorous lost by vegetative production. Nuclear seedlings restores vigor lost by vegetative propagation. The some amount of vigor is lost in vegetative propagation, so nuclear seedlings restores it. It increases probability of survival under varied condition. It increases survival under unfavorable conditions. Virus-free clones of citrus species is the only practical approach of production of seedling. They have also produced virus-free seedlings in citrus approach in citrus species. Disease-free plants are obtained. The same means disease-free plants are obtained. So these are the main significance, or you can say advantages of polyembryony. Now moving on, what is the main drawback? There is one very major drawback of polyembryony. You should know it, and that drawback is it interferes with hybridization programs. That is the major drawback. Now what is the classification? How is polyembryony classified? Number A, that is based on frequency. There are three types. First one is strictly polyembryony. Second one is nearly polyembryony, and third one is polyembryonic. In strictly polyembryony, there is polyembryony of up to less than 7%. In nearly, it is up to 6 to 10%, and in polyembryonic, it is more than 10%. Now in B, it is also based on genetic composition. It is of two types: gametophytic and sporophytic. So these were the drawback and classification of polyembryony. So hope you understand all the concepts of today's lecture. For any queries, do comment. If you like it, please press the like button. Do share and subscribe. Keep making notes. That's all for today. Have a nice day. Thank you.